Introduction to the Biology of Friendship. Hello and welcome to our video on the biology of friendship. Friendship is a complex and multifaceted phenomenon that has been studied by scientists and philosophers alike for centuries. But what is it about friendship that makes it so unique and essential to our well-being? In this video, we'll explore the biological underpinnings of friendship. From the release of oxytocin during social bonding to the neural mechanisms of empathy, we'll examine the fascinating biology behind friendship and discuss the implications of this research for our understanding of human relationships. The role of oxytocin in social bonding. One of the key hormones involved in social bonding is oxytocin. Often referred to as the cuddle hormone, oxytocin is released during physical touch and social interaction, and has been shown to play a crucial role in the formation and maintenance of friendships. Studies have found that oxytocin levels increase during social bonding activities such as hugging, kissing, and sex, and that this hormone helps to reduce stress and anxiety while promoting feelings of trust and relaxation. But oxytocin is not the only hormone involved in social bonding. Other hormones such as dopamine and vasopressin also play important roles in the formation and maintenance of friendships. Dopamine, for example, is involved in the reward system of the brain and is released during pleasurable activities such as eating, sex, and social interaction. Vasopressin, on the other hand, has been shown to play a role in the formation of long-term social bonds, and has been implicated in the development of monogamous relationships. Neural mechanisms of empathy and friendship. So what happens in the brain when we form friendships? Research has shown that the neural mechanisms of empathy and friendship are complex and involve multiple brain regions. The mirror neuron system, for example, is a network of brain cells that fire both when we perform an action and when we observe someone else performing the same action. This system is thought to be involved in empathy and social understanding, and is active when we form friendships. Other brain regions such as the prefrontal cortex and the anterior cingulate cortex are also involved in the formation and maintenance of friendships. The prefrontal cortex, for example, is responsible for executive function, decision-making, and social behavior, while the anterior cingulate cortex is involved in error detection, conflict monitoring, and emotion regulation. The impact of stress on friendships. Stress is a natural part of life, and can have a significant impact on our friendships. Chronic stress, for example, can lead to the release of stress hormones such as cortisol, which can suppress the immune system and lead to a range of negative health outcomes. Stress can also interfere with our ability to form and maintain friendships by reducing our levels of oxytocin and other social hormones, and by impairing our ability to engage in social behavior. Conclusion and future directions. In conclusion, the biology of friendship is a complex and multifaceted phenomenon that involves the release of oxytocin and other social hormones, the activation of neural mechanisms such as the mirror neuron system, and the involvement of multiple brain regions. By understanding the biological underpinnings of friendship, we can gain a deeper appreciation for the importance of social relationships in our lives, and develop new strategies for building and maintaining strong, healthy friendships.